guys, I wanted to film a quick video for you about what we went over in class today because I know we were super rushed. So I want to review for you guys DNA structure and go over some specifics about DNA replication, like how do you actually do the complementary base pairing, stuff that was not covered in our replication fork video. Make sure you go and review that replication fork video because you definitely are going to need to know the parts of the replication fork and how the leading and the lagging strand synthesis, how that's totally different. Okay. What I want to go through really quick, sorry, I don't think my board is dry. I just washed it. But I wanted to go over DNA structure with you guys. And I'm going to start by going over the structure of the nucleotide. And so remember that DNA is one of our macromolecules. It's called a nucleic acid. And that nucleic acids are made up of subunits called nucleotides. And so our nucleotide has a phosphate here. This phosphate represents the 5' end of the nucleotide because it's attached to the 5th carbon in this sugar ring right here, okay? This is the 3' end of the molecule. This is the molecule that's on the 3rd carbon in the ring of the sugar. So that's where we get the 5 and the 3 from. The sugar is also attached to the nitrogenous base. And if you remember, there are 4 different kinds of nitrogenous bases. We have adenine, guanine, cytosine, and thymine. And those are going to be the bases that make up our DNA strands. The differences between adenine, guanine, cytosine, and thymine, they have some structural differences. For example, these two, adenine and guanine, are what we call purines. If you remember that word purine, it's because they have 2 rings in their structure, and then cytosine and thymine are what we call pyrimidines. And they have 1 ring in their structure. So we have a couple structural differences here of our nitrogenous bases. But a nucleotide is always going to have the same 3 parts: phosphate, sugar, nitrogenous base. And that nitrogenous base could either be adenine, guanine, cytosine, and thymine. The nitrogenous bases are the ones that stick out in the middle of the DNA strand. So let's do like an untwisted DNA strand up here. So my backbone here, this would be my phosphates and my sugars, and they alternate. It goes phosphate, sugar, phosphate, sugar. So this is the phosphate of one nucleotide. This is the sugar of the same one, phosphate, sugar, phosphate, sugar. And then the rungs of the ladder, those are our nitrogenous bases. Those are our adenine, guanine, cytosine, and thymine. And remember that because DNA is double-stranded, we've got A, complementary base pairing with T, and there's 2 hydrogen bonds holding them together, and G, complementary base pairing with cytosine, or C, and we've got 3 hydrogen bonds holding them together. Okay, so then we've got to talk about how do our nucleotides polymerize or go into a strand. So the 3' end of one nucleotide, I'm just going to draw that smaller so I have a little more room. It's going to attach to the phosphate. So remember, the phosphate is the 5' end of the next nucleotide down. So let me draw that real quick, and then I'll make sure that it gets on the board. There you go, like that. So here we've got the 3' sugar of this nitrogenous base making a bond with the 5' phosphate of this nitrogenous base, and we call that bond a phosphodiester bond. Phosphodiester bond. And then if you remember, our nucleotides are held together, complementary base pairs are held together with hydrogen bonds. So you've got 2 different types of bonds to really worry about. You've got hydrogen bonds, which are holding together the nitrogenous bases of the 2 strands of DNA. So like if we're going to do an untwisted ladder here, there's my hydrogen bonds holding my base pairs together. And then we have phosphodiester bonds. These are the bonds that are found in the backbone of the DNA strand, and they hold the sugar from one nucleotide to the phosphate of the other nucleotide, or the one below it. And so this allows us to have this double-stranded double helix, for lack of a better term. And we remember that one of the important things to know about our double helix is that it is anti-parallel. So if this end is my 5' end here, this strand would end in 3' down here. If this is my 3' end of this strand, then it ends in 5' down here. They're anti-parallel. Remember that we talked about anti-parallel means they run in opposite directions. So they're parallel lines, but they run in opposite directions. And this is, of course, what Watson and Crick discovered about the structure of DNA that led them to then understand the function. If you guys remember, I talked to you about how the last line of their paper says something along the lines of, now that we know the structure, we also know the function. And they went on to produce what we call the central dogma of molecular biology, which discusses how DNA actually works. So let me get some fresh board space here, and then we can talk a little bit more about the central dogma and the first step of central dogma, which is DNA replication. I want to leave my DNA diagram up here for a second. I'm going to put a little more detail in it so that we can see what's going on. Oops, did you see my mistake? I put it on the wrong side. Okay. Then we've got nitrogenous bases sticking out here and on this side. There we go. All right. So, central dogma. Francis Crick. Francis Crick says that now that we know the structure of DNA, the function becomes apparent, which means that the structure tells us how it functions. And since DNA is a double helix and it has 2 strands, we know that it can replicate. And so that means that the strands can pull apart and they can be used as a template to make a new strand of DNA. So let's go over that central dogma, and then we'll talk a little bit more about replication. So we've got the central dogma of molecular biology. It is the set of guidelines that all of molecular biology is based around. That's why it's called a dogma. And it goes something like this. It says that DNA, change markers real quick. I think my board's still a little bit wet. There we go. DNA can make more copies of itself through a process called replication. DNA can make RNA through a process called transcription. And remember, we talked about how DNA and RNA are like dialects of the same language. So this would be like Northern English and Southern English. And so when we're going from north to south, we have to transcribe. We're not going into a different language. We're just changing the language. We also talked about how that's kind of like transposing when you're in music. Like if this was in the key of B flat and you wanted to go to the key of C, then you would transpose it. It's the same language, just a different key. So RNA is then going to be translated into a protein. And remember that proteins build our cellular structures. So all of your cellular structures basically, except for your cell membranes, are made out of protein. And so this is how the message to make all of our cells is encoded into DNA. So the sequence of the letters of DNA, the A's, C, T's, and G's, will tell us the sequence of the letters in RNA, and then that will tell us the amino acid sequence in the protein. This section is called translation because RNA is a nucleic acid and a protein is a protein. And RNA and proteins, they're different types of biological molecules. Remember, we had 4 biological molecules that we learned at the beginning of the year, and nucleic acids and proteins were 2 of those. So that's why this process is called translation, because we're going from one language to another language completely. And so that's kind of a graphical representation of the central dogma, and it makes it really easy to think about how DNA can make more of itself through replication. DNA is transcribed into RNA, and then RNA is translated into proteins, and that's how we make everything in our cells. These proteins could be enzymes that would go and make other things happen. They could be structural proteins, like proteins that build our cells. They could be signal proteins. There's a plethora of things that they could be, but this is all how they're made. So notice that over here in my DNA diagram that I drew it in 2 different colors. That's because replication occurs in what we call a semi-conservative manner. And that semi-conservative manner means that the parent strand is going to split and be replicated into two daughter strands. So I'm going to do like a little representation of it, and then we'll start talking about the letter part. So if this is my parent DNA, here's my 5 prime, there's my 3 prime, here's 3 prime, here's 5 prime. So cells do DNA replication in S phase of the cell cycle, if you guys remember that. And that in S phase of the cell cycle, we're going to make an identical copy of DNA. This ensures that the 2 resulting daughter cells will be identical because their DNA is identical. So how does that identical thing happen? Well, it happens through what we call semi-conservative replication. So when replication occurs, helicase is going to split the parent DNA apart. So I'm going to say that this arrow is helicase, just for example's sake. Helicase splits the parental DNA apart. So there's my 5 prime strand, 3 prime. Put a lot of space here because remember it's split apart in the replication bubble. 5 prime. And then my DNA polymerase 3 is going to come in and make a copy of this. Like that. So where I had one parent strand here, now I have 2 identical daughter strands. And these 2, this one would go into one cell and this one would go in the other cell during mitosis. And now these 2 cells would be identical because they have identical copies of DNA. We call this model semi-conservative, and I know in class we talked about what semi-conservative meant, but I want to go over it again. Semi-conservative. And that means that when we do DNA replication, the 2 daughter strands are half old parent DNA and half new daughter DNA. So that's what we mean by semi-conservative. And that's what ensures that the message is conserved in the DNA so that when we do cell division, our cells end up to be identical because this old strand of DNA was replicated into this one. This old one was through complementary base pairing was replicated into this one. And now these 2 resulting daughter strands say the exact same thing as the parent strand did. So that's why we call semi-conservative replication. Now let's put some actual letters to this semi-conservative replication. So if I give you a parent strand of DNA, here we go, parent strand. And I'm just going to do one side of the parent strand just for time's sake. So let's say that this is my parent strand. Like that. It's a really short strand of DNA, but we're just doing it for example's sake. DNA polymerase 3 is going to come by and read this strand and do complementary base pairing, and A is going to pair with T, G is going to pair with C, and we're going to end up with a strand, a daughter strand that looks like this. So notice that my new DNA that was laid down is a complement to my old DNA, or what we would call this the template strand, and this is the new strand. So here's AT, TA, AT, CG, GC, GC, CG, TA, AT, CG, AT. I think you guys get it. So we're just using complementary base pairing. So if I were to have if I were to give you a sequence of template DNA, I expect you to be able to fill in the opposite side so that this whole thing is of course going to be the daughter DNA, but this was the template strand and this was the new strand. So I expect you, if I give you a template, I expect you to be able to fill in the daughter strand side right here, okay? That's what, as far as replication goes. I also expect that you know the replication fork. Let me grab that off my desk real quick. There we go. So again, the replication fork, I know I made a video about this earlier, but I just wanted to remind you what the replication fork looked like. So it'll remind you to go watch that video, okay? Watch the video. Let me know if you need anything else with DNA structure. We've got to move on to transcription and translation tomorrow, but I'll be more than happy to help anybody out as they have any questions. So replication, remember, it's the first step of the central dogma. It's done in a semi-conservative manner. And if we have a template strand, we should be able to fill in what the daughter strand looks like given that we know about complementary base pairing, A's pair with T, G's pair with C. One more thing. I know I've said one more thing 1,000 times, but remember that helicase unwinds or unzips the DNA strand at an AT-rich sequence, and that's very significant because if you remember the difference between our AT pairs and our GC pairs, A's and T's are held together with 2 hydrogen bonds, G's and C's are held together with 3 hydrogen bonds. This one's easier to break apart than this one, which is why DNA helicase begins replication at an AT-rich sequence. And by AT-rich, we mean it's got a lot of A's and T's in it. So that whole 2 bonds versus 3 hydrogen bonds thing, that's really important, okay? So DNA structure, it's anti-parallel. The backbone is held together with phosphodiester bonds. The 2 strands are held together with hydrogen bonds. There's a different number of hydrogen bonds in between each pair, and we have to be able to label 5' and 3', okay? Also, you need to know the difference between your purines and your pyrimidines, and of course, complementary base pairing. I hope this video helps. We will discuss a little bit more about this tomorrow. I hope you guys have a good night.